Now let me introduce our speaker today, Jordi Alcaraz, Associate Researcher at IBEC. Jordi Alcaraz graduated in physics at the Universitat de Barcelona and obtained his PhD in biophysics in the fields of cellular biophysics and bioengineering in 2002. During his postdoc, he pursued research aiming to understand how biophysical cues from the tissue microenvironment control differentiation and cancer progression at the single-cell level. Currently, he is a Hunter Associate Professor of the School of Medicine and Health Science at the UB since 2012, where he leads the Translational Lung Mechanobiology group. He is also a researcher at the Functional Unit of Thoracic Tumors at the Hospital Clínic of Barcelona and of the Spanish Respiratory Disease Networking Biomedical Research Center, CIBER. His research line is focused in the study of how the microenvironment controls cells' behavior and misbehavior in normal and disease conditions, particularly in lung cancer and fibrosis. Their goal is to define new therapeutic strategies against these diseases, as well as to identify novel biomarkers. For this purpose, the group works in close collaboration with clinical groups and often use primary tissues, cells from patients. Jordi, thank you for being here today. The screen is yours.
Thanks, Christina, for your introduction, and all of you for showing up this Friday morning. And this is the first seminar that I give at IBEC since I was appointed as an associate researcher. And I would like to take this opportunity to give an overview of the kind of translational research that we do in our lab, and also to highlight the ongoing collaborations that we have with other groups at IBEC. So we work in lung cancer. My lab is located at the School of Medicine at the University of Barcelona. And we are experiencing exciting times in tumor biology research, because we are facing a paradigm shift in which solid tumors are increasingly regarded as abnormal organs. And this is an important shift because it highlights the fact that cancer cells actually need a lot of help from the surrounding connective tissue or stroma for growth and dissemination. And this idea is illustrated in this histologic section, which that corresponds to a lung adenocarcinoma, in which you see here a cluster of cancer cells that is surrounded by by a layer of this stroma or connective tissue that is rich in activated fibroblasts here in red. And it's this interaction, this crosstalk, that is critical for the tumor growth and dissemination. And fibroblasts in particular are very efficient in helping the logistics for this tumor growth and dissemination. And accordingly, there is a growing translational interest in studying TAFs, that or what we call tumor-associated fibroblasts, or others call them cancer-associated fibroblasts. This, of course, has therapeutic implications, because it means that in addition to targeting cancer cells, which has been the traditional approach during the last 40 years, there is a growing interest in targeting also these TAFs or other stromal cells. Of course, this makes the picture much more complex, because it's no longer enough to care about cancer cells, but it's also relevant to care about the interaction that cancer cells have with TAFs, but also with immune cells, endothelial cells, and other stromal cells, and also with the extracellular matrix that these cells, particularly TAFs, deposit on the tumors. And we've been working on this in this field. And we've done a lot of work on the interaction between cancer cells and TAFs that has been published in different papers shown here. And more recently, we started analyzing how these TAFs interact with immune cells and with endothelial cells. So to move on, I need to give you a little bit of background. And let me start by saying that fibroblasts in normal conditions, they are quiescent, and they deposit little matrix. But in different conditions, like in cancer or in fibrosis, and also during repair, these fibroblasts become activated. and they acquire a fibroblast that it's called a myofibroblast or activated phenotype that is characterized roughly by the intracellular expression of alpha-smooth muscle actin, which is an actin isoform that provides these cells with a strong contractile properties similar to muscle cells. And at the same time, these cells become very efficient in depositing large amounts of extracellular matrix, particularly collagen 1. And this transition is driven by different cytokines, particularly transforming growth factor beta 1 is very efficient in activating fibroblasts. And also there is growing evidence that matrix stiffening itself is also a contributor to this activation. Lung cancer is it's a very important disease because it remains the first cause of cancer-related death worldwide, and it has a very suboptimal survival rate of only 18% 5 years after diagnosis, which is much worse than other prevalent cancer types like breast or prostate. And we've been working in the two most common histologic subtypes of lung cancer, which are adenocarcinoma, that typically arises in distal sites of the lung, and also squamous cell carcinomas, which arise in the more proximal sites of the lungs and is strongly associated with smoking. And we also have done some work on large cell carcinomas, which are much less frequent, but they are very aggressive, and its tumor biology remains very unknown. So how do we study these staph? And we do so by using a variety of preclinical models that I would like to briefly describe to you. The first models are the in vitro and are based on patient-derived TAFs obtained from surgical lung cancer patients. And from the same patients, we also obtain fibroblasts that are far away from the tumor that we use as controls. And we have been able to obtain these TAFs in collaboration with the lung oncologist, Dr. Noémie Reward, and Dr. Gascon at the hospital clinic. And actually, we were the first group to create this collection of TAFs, of lung TAFs in Spain. And we culture them either alone or with cancer cells or immune or endothelial cells into the or through the conditions. We also use tumor xenografts that are based on co-injecting cancer cells or fibroblasts into the back of immunodeficient mice, and then monitor tumor growth and also their dissemination to the lungs and after several weeks. And this is these are two examples that illustrate the contribution of fibroblasts. So when you co-inject fibroblasts with cancer cells, shown here in blue, you get bigger tumors and more metastases to the lungs compared to injecting cancer cells alone, shown here in black. And finally, I would like to introduce you a third preclinical model that is new, and it's that we call it Ex-Vivo, and it's based on isolating a piece of a fresh tumor from a surgical patient, and then slicing up this fresh piece of tumor into thin sections that we lay on a multi-well, and with a culture medium that has been optimized to preserve the integrity and the function of these tissue sections for a few days. And this provides a time window that allows you to add different drugs at different concentrations and see the direct response of these tumor sections on these drugs. And this Ex-Vivo assay has different advantages, particularly that is very fast, and it allows checking the response of the same patient to different drugs. And we use it to identify new or useful drugs against staph. And we also do that in collaboration with a variety of companies that have promising drugs in this arena. And this is just to illustrate how this assay works. So this is the response of these tissue sections to an anti-fibrotic drug. This is the staining of collagen. And you can see that over time, the amount of collagen decreases compared to control. So what I would like to tell you next, it's summarizing here. So basically, what we have learned about what's wrong about these lung tufts, also if we can fix these alterations, and finally, to introduce you different new tuft-related markers that are useful for diagnostic and prognostic purposes. So let's begin with the first part. And in here, I will briefly mention 3 stories that are related to tasks from each individual histologic subtype. So let's begin with the excessive fibrosis that we identified in adenocarcinoma tufts. Remember that adenocarcinomas are the most frequent lung cancer subtypes. And when we started working with tufts a few years ago, one of the things we did was to analyze their epigenetic landscape in collaboration with the group of Manel Estaller. And to do that, what we did was DNA methylation profiling of tufts and control fibroblasts from the same patients. And what we found is that lung tufts, shown here in blue, they cluster together. And the control fibroblasts shown here in yellow, they also cluster together. And we got a bunch of genes, more than 700 genes that become differentially methylated in tufts compared to control fibroblasts. And this means that these tufts are epigenetically reprogrammed. And most of the genes go from red to green. This goes actually like the traffic light. So red means that these genes are enriched in methylation, in DNA methylation, and this acts as a repression for transcription. And when they go to green, it means that they lose methylation, and therefore they become accessible for transcription. So most genes go from red to green, but then there is a subset of genes that go the other way around. They go from green in control fibroblasts to red in tufts. And these are the genes that typically draw the attention of researchers because intuitively we suspect that the genes that become repressed epigenetically in tumors are genes that may be annoying to the tumor, and therefore they may contain tumor suppressors. And we were very surprised to see that the top methylated gene in tufts was SMAD3. And the reason why this was surprising is because SMAD3 is an important transcription factor for fibroblast activation. And I just told you that tufts are activated in tumors, and yet it appears to be that an important transcription factor for such activation is epigenetically repressed. So we decided to look more in depth into this paradoxical observation. And actually, the SMAD3, it's part of what it's called the canonical TGF-beta pathway, in which upon binding of TGF-beta binding to its receptors, they activate through phosphorylation SMAD3 and its closely related homolog SMAD2 that together with SMAD4 translocate to the nucleus to activate the expression of fibrosis genes like alpha-SMA or collagen. So we decided to look at what's going on with SMAD2 and SMAD3 in our tufts. And in vitro, when you stimulate with TGF-beta these tufts from squamous patients, you get a weak SMAD3 activation, which is consistent with the epigenetic repression that I just showed you before. And as a compensation, you get a strong SMAD2 activation. And you see the same behavior in tufts from other squamous patients. Conversely, and surprisingly, when you do the same experiment with tufts from adenocarcinoma patients, you see a strong SMAD3 activation and a weak SMAD2 activation. And that was surprising because SMAD3 was supposed to be epigenetically repressed in adenocarcinoma tufts as well. So we reanalyzed more carefully the methylation of SMAD3 in tufts, and we found that it's higher than control fibroblasts in both adeno, shown here in blue, and the squamous tufts, shown here in red. But the methylation of SMAD3 is much higher in squamous tufts compared to adenocarcinoma tufts, revealing that the methylation of SMAD3 in adeno tufts is not sufficient for its transcriptional repression, but it is sufficient in squamous tufts. We then wonder why there is more SMAD3 methylation in squamous tufts compared to adenocarcinoma tufts. And we wonder if this differential methylation could be related to smoking, because I told you before that squamous tufts, they arise in more proximal airways and are strongly associated with smoking. So one may expect that these tufts are more directly exposed to cigarette smoke particles. And to address this question, we did the following experiment. We use this assay that allows you to condensate particles from smoking cigarettes into the culture medium, and then use this culture medium enriched in smoking particles to stimulate normal fibroblasts. And what we found is that over time there is an increase in SMAD3 methylation caused by this medium enriched in smoking particles, but there is no change in the methylation of SMAD2, revealing that for whatever reason, smoking particles are able to methylate specifically SMAD3. Now, I told you before that SMAD3 is supposed to be a transcription factor that is very important for fibroblast activation and expression of fibrotic genes. And therefore, one should expect that there is more fibrosis in squamous tufts compared to adenocarcinoma tufts. And we test this hypothesis first in vitro. And I show here a panel of fibrosis markers like alpha-SMA, collagen, or tissue beta. They're consistently highly expressed in adenos compared to squamous, shown here in red. And likewise, in patient samples, you get more collagen deposition in adenocarcinoma patients compared to squamous patients. And you even get more collagen deposition in tumors from never smokers, in agreement with the idea that the smoking represses SMAD3 and therefore fibrosis. These observations have also an interesting translational potential, because they suggest that if adenocarcinomas are more fibrotic, then they should respond better to antifibrotic drugs. And we've been able to address this question in collaboration with a pharmaceutical company that developed an antifibrotic drug called nintedanib that reported a clinical survival benefit in a large clinical trial with thousands of patients in adenocarcinomas, but not in squamous cell carcinoma patients. And actually, because of this data, this drug became approved to treat lung adenocarcinoma tumors. But they didn't know why this drug did not work in squamous carcinomas. And at that time, at the time that this clinical trial was published, we already had some insights of the differential fibrosis in adenotufts compared to squamous tufts. So we started a collaboration with this company to see if tufts were underlying this differential response to this antifibrotic drug. And in vitro, we show that actually that is the case. So the expression of different fibrosis markers like collagen-1, it drops more efficiently with increasing doses of the drug in adenocarcinoma tufts, shown here in blue, compared to squamous tufts, shown here in red. And likewise with alpha-SMA, you see a more efficient downregulation with the drug in adenos compared to squamous. The question was, what was the underlying mechanism? And we wonder if SMAD3 could account for this differential response to this antifibrotic drug. And we did a bunch of experiments to address this question in vitro and in vivo. And here I'm just showing the in vivo data in which we co-injected cancer cells with fibroblasts that were either SMAD3 wild type or SMAD3 deficient. And when using SMAD3 deficient fibroblasts, you get no response upon treatment with nintedanib of these mice upon a few weeks. And you see similar amounts of collagen deposition and similar activation of proliferation shown here by K67 staining. In contrast, when you co-inject SMAD3 wild-type fibroblasts with these cancer cells, you get strong tumor growth and a strong collagen deposition and Ki-67 staining. And conversely, the mice treated with the drug, you get a strong suppression of tumor growth, shown here, that is associated with a marked drop in the collagen deposition and Ki-67 staining. So this identified a new resistance mechanism to this antifibrotic drug based on the SMAD3 epigenetic repression in TAFs. To summarize this first part, I show you that in adenocarcinoma TAFs, because of their anatomical location far away in distal sides of the lung, they get a weak exposure to cigarette smoke particles, which elicits a weak promoter methylation of SMAD3. And this means that upon activation with TGF-beta, the response is dominated by SMAD3, and this elicits strong fibrosis and a strong response to nintedanib and possibly other antifibrotic drugs. At the same time, this data provides a rationale to use antifibrotic drugs in adenocarcinoma patients that are treated with radiotherapy, because radiotherapy, as a common side effect, elicits fibrosis, and this actually becomes a very problematic side effect that may threaten the health status of these patients. And we are analyzing this possibility with clinical collaborators at the hospital clinic. Conversely, in squamous carcinoma TAFs, you see the other way around. Because of their anatomical locations in the proximal airways, they have a higher exposure to cigarette smoke particles that elicits a strong SMAD3 methylation, and this means that when exposed with TGF-beta, they get a high response through SMAD2 rather than SMAD3, and this elicits a weak fibrosis and a weak response to antifibrotic drug. And this means that for squamous carcinoma patients, other drugs are required to treat TAFs, and this is an active area of research in our group nowadays. So let's move to the next story. Following with the abnormalities of squamous TAFs, one of the things that we did in collaboration with Ana Laverdadi at IBEC was to characterize the contractility of TAFs in vitro in response to TGF-beta. And we found that adenocarcinoma TAFs exhibit similar contractility to control fibroblasts, so they are normal from this point of view. But the squamous TAFs are not. They are actually much more contractile. Even they exhibit a 50% higher traction forces. And we are currently expanding these experiments in collaboration with Annalisa Calo at IBEC. And this abnormal mechanobiology was associated with abnormal signaling of important mechanobiology players shown here, because these squamous TAFs, they exhibit an upregulation of expression of beta-1 integrin and downstream activation of FAC, shown here. It's also higher in squamous TAFs, and also they exhibit a higher activation of ERK. And this illustrates that squamous TAFs have an abnormal mechanobiology and therefore could be more responsive to mechanobiology drugs, particularly to FAC inhibitors. We are also trying to examine this possibility. On the other hand, we have analyzed angiogenesis in these tumors, and we have learned that angiogenesis is lower in squamous carcinomas compared to adenocarcinomas, and this is associated with a drop with a lower expression of angiogenic markers, shown here in red compared to blue, like VEGF-A or CD31, shown here at the RNA level, and you get also a downregulation of these pro-angiogenic factors at the protein level. And because fibroblasts are very efficient in regulating angiogenesis, actually in promoting angiogenesis, we wonder if TAPs could underlie this differential angiogenic potential. And to address this question, we used an in vitro assay in which you culture endothelial cells on top of matrigel, and after 16 hours, they deposit this structure. They make this network structure that resembles a rough vasculature that you can quantify. And we did these experiments by using conditioned medium of TAPs from adenos and squamous patients activated with TGF-beta. And what we found is that when using conditioned medium from adenocarcinoma TAPs, you get an increase in this network formation, but you don't see this increase when using conditioned medium from squamous TAPs. Again, supporting that TAPs may underlie the lack or the poor angiogenesis in squamous carcinomas. We confirmed these observations, actually the implication of SMAD3 in these observations, and we examined the expression of CD31, which is a marker of endothelial cells, in the tumor xenografts that I showed you before, in which we co-injected cancer cells with SMAD3 wild type or deficient fibroblasts. And we found that with the SMAD3 deficient fibroblasts that behave like squamous TAPs, you see a less expression of CD31, meaning or confirming that in vivo these fibroblasts are less able or less efficient in promoting angiogenesis. We also have evidence that associates this poor angiogenic potential in squamous TAPs with its abnormal mechanobiology signaling, and we are currently studying this interaction. And to finish this block, we'd like to tell you what we have learned about the abnormalities of TAPs from large cell carcinoma patients. And these abnormalities have to do with senescence. Now, senescence is the word used to refer to cellular aging, and senescence is characterized by permanent growth arrest, but yet senescent cells remain metabolically active, and this metabolism drives the expression and the secretion of many factors. And actually, this secretory phenotype has been associated with cancer aggression because senescent TAPs have been reported in a growing list of aggressive cancer types like breast, oral, liver, and ovarian cancer. And we were the first ones to identify an enrichment in senescent TAPs in larger carcinomas, shown here by the positive staining of standard senescence marker, which is the beta-galactosidase that stains in blue senescent cells. And we found an enrichment in these senescent cells in TAPs from larger carcinoma patients compared to squamous adenocarcinomas or control fibroblasts. We also confirmed that these senescent TAPs, they contribute to the aggression, to the aggressive nature of these tumors by doing a bunch of in vitro and in vivo experiments. Here I'm just showing the in vivo data in which we co-injected larger carcinoma cells with fibroblasts that were either normal or meaning non-senescent or senescent. And over time, what we found is that the tumors bearing senescent fibroblasts, shown here in red, they make bigger tumors and more metastases to the lung compared to tumors bearing non-senescent fibroblasts, shown here in blue. And in both cases, they are much bigger and make more metastases than just injecting cancer cells alone. Now, this data is important because it's the first piece of evidence that provides a potential mechanism of why these tumors are so aggressive, and by identifying these senescent TAPs. And we also wonder why these TAPs are senescent in large carcinomas specifically. And we wonder if cancer cells were involved in this senescence induction. And to address this question, we did the following experiment. We use transwells to co-culture cancer cells, grown here in the insert, with normal fibroblasts shown cultured at the bottom of the transwell. And over a few days, what we found is that these fibroblast became senescent only when they were co-cultured with larger carcinoma cell lines, but not when they were co-cultured with cancer cells from other histologic subtypes, revealing that these larger carcinoma cells, they secrete factors that induce paracrine senescence in the fibroblasts. And we wanted to know what these factors could be. And to address this question, we did expression array analysis in which we found that the expression of profiles of larger carcinoma cell lines is different from cell lines from other histologic subtypes. And through bioinformatic analysis, we identified MMP1 as a gene that encodes for a secreted protein. It's a metalloproteinase that degrades collagen, that is heavily upregulated in larger carcinoma cells compared to non-larger carcinoma cell lines. And we decided to test the role of MMP1 in this senescence induction. And for that purpose, we repeated the co-culture by using here larger carcinoma cells that were either MMP1 wild type or deficient. And what we found is that using MMP1 deficient cancer cells, we did not see the increase in beta-gal positive fibroblasts using different plasmids for MMP1 downregulation and also different larger carcinoma cell lines. Whereas when co-culture with control cells, you got an increase in the amount of senescent fibroblasts as expected. We also did an in vivo validation by injecting MMP1 wild type or deficient larger carcinoma cells, and found that knocking down MMP1 was sufficient to elicit smaller tumors, and also lower metastases to the lungs, and also lower amount of senescent fibroblasts shown here with this histologic analysis of a senescent marker called centragore, compared to that is high in control tumors, but is much lower in tumors with MMP1 knockdown cancer cells. So this data is nice because it identifies a completely new function of MMP1, which is to induce senescence paracrinely in fibroblasts. That is very different from its traditional function, which is to degrade collagen. But this data also has translational potential because it reveals that you could target these senescent TAFs as a new therapeutic approach in these tumors, and you can do that with drugs called senolytics. And we started working in this arena by testing the role of a senolytic drug called ABT737. And in vitro, you can see here that it downregulates the number of senescent fibroblasts compared to control through our increased apoptosis. And we are doing this analysis in collaboration with Joan Montero at IBEC. And at the same time, this drug reduces markedly the ability of the condition medium of these senescent TAFs to enhance growth of cancer cells, as shown here. And to finish the last part of the talk, I would like to briefly describe you the work that we're doing on identifying new diagnostic and prognostic biomarkers associated with TAFs. In the first work we'd like to describe to you, we realized that it was unknown the prognostic value of standard TAF activation markers like alpha-SMA and collagen. And we managed to do this study in collaboration with Fiberres, in which we managed to analyze the expression of alpha-SMA in histologic sections and show here, and collagen through serous red staining that allows you to see in bright field or even the fibers of collagen filaments through polarized light in a large cohort of more than 200 patients. And with these stainings, we did an image analysis in which we assess the percentage of positive staining area for each marker. And then we stratify the patients into groups, the groups that have higher expression and lower expression for each marker. And what we found is that the group with high expression of alpha-SMA, shown here in blue, these are survival plots, they exhibit a worse survival than the group with lower expression values. And likewise, we got similar results when analyzing cirrus red by bright field or cirrus red by polarized light. And what was interesting was to see that when we computed the hazard risk associated with these markers, which tells you the increased risk of death provided by this marker, and correcting by the current gold standard of patient staging, which is based on TNM. T stands for the tumor size, N for the nodal status, and M for the metastasis. We found is that the three markers were statistically significantly associated with increased risk of death. This means that surgical patients that exhibit higher levels of these markers would benefit for a closer follow-up based on these new markers associated with TAFs. We are currently expanding this analysis by analyzing the architecture of these collagen filaments that we can obtain by polarized light imaging of this collagen staining and using advanced algorithms, and that allow you to grow these filaments here and from them assess the length, the width, or the relative orientation. And to give you an example of the kind of data we are getting, we found that the width of the collagen filaments in adenocarcinoma patients, it does have prognostic value. And this illustrates a new example of novel biomarkers associated with collagen, with the collagen architecture that we are doing in collaboration with Enrico Almici at the IBEC, in the group of Josep Samitier. And the last project I would like to tell you, it's a brand new project that we are doing in collaboration with Antivis, that is a Swiss company that has developed an atomic force microscope that is has been optimized to measure the mechanical properties of tumor biopsies. And the idea of this project is to obtain lung cancer biopsies with a core needle. These biopsies look like a cylinder, and use this device to measure the to obtain force curves, many force curves on the surface of this biopsy, as much as 10,000 force curves, and from each one you can obtain information on the different mechanical parameters. And so, because we got so many data values from each patient, this allows you to use the tools from big data. And you can do this acquisition very fast, within 3 hours. And the idea is that you can obtain nanomechanical signatures that could help diagnose better these tumors, and much faster than any other diagnostic techniques that are used currently in the clinic. And also in the future, we plan to check if any of these nanomechanical signatures can help predict the response of these patients to treatments that are available, so that in the future it could help guide what is the best treatment option for each patient. So in summary, I show you that lung TAFs are all activated in vivo, but yet they show the different phenotypic alterations depending on the histologic subtype. And these histologic specific alterations provide translational opportunities. For instance, the robust fibrosis in adenocarcinomas, they support the use of antifibrotic drugs in these tumors. The aberrant mechanobiology in squamous carcinomas, they support the use of mechanobiologic drugs like FAK inhibitors. And the normal enrichment of senescent TAFs in large cell carcinomas supports the use of senolytic drugs in these tumors. I also told you a new function of MMP1, which is to induce paracrine senescence. And finally, I give you examples of how markers of activated TAFs or collagens may be useful for diagnostic or prognostic purposes in lung cancer. And I would like to finish by thanking the postdocs and graduate students in my group and my other collaborators, particularly Dr. Reward at Hospital Clinic and Dr. Rodisky at the Mayo Clinic, and also my funding sources. And I'm happy to take